So we had spent the first part of this chapter talking about the innate system. The innate system is what you're born with. You don't have to do anything to get it. It's in place, and the characteristics of it are that it acts the same every single time, regardless of the pathogen that it encounters, and most importantly, it's not specific. So now what we want to do is we want to talk about part two, which are our adaptive defenses. So this is our specific defense system that eliminates most of the pathogen and abnormal cells in our body. So it does this by amplifying the inflammatory response, by activating complement. The shortcoming to this is that it has to be primed by initial exposure to that antigen or that foreign substance, and that priming takes time. So characteristics of our adaptive immune system is that it's specific, right? It's going to recognize and target specific antigens, unlike our innate immune system that targets all antigens. It is systemic, so it's not restricted to the initial site like we saw with the innate system. It has memory, which means that basically after you have a confrontation with a known antigen, it makes a little memory chip and it stores that away. So every time you're exposed to that again, you don't get sick. It knows exactly what to do. And so that idea is the basis for immunizations that we will talk about as well. There's two main branches of the adaptive system. There's the humoral, which is also referred to as antibody-mediated immunity, or AMI, and cellular or cell-mediated immunity, which is referred to as CMI. So let's talk about humoral first. Humoral is going to rely on antibodies. So antibodies are chemicals that are produced by your lymphocytes that circulate freely in the body fluid. They bind temporarily to a target, they temporarily inactivate it, and they mark it for destruction by the phagocytes or by complement. Humoral immunity has extracellular targets. In other words, it's going to target things that are not inside of your cells, but everywhere else outside of the cells. Cellular immunity is when the lymphocytes are going to act against a cell that has been targeted. They're going to act directly by killing that infected cell, or indirectly by releasing chemicals that enhance the inflammatory response or activate other lymphocytes or macrophages. Cellular immunity has cellular targets, so it's going to go after cells that the pathogen is now inside. So let's start with talking about antigens. So antigens are substances that mobilize your adaptive defenses and provoke an immune response. Basically, they are anything that can make you sick, essentially. All right. They are the target of all of our adaptive immune responses. Most of them are large, complex molecules that are not normally found in the body, like bacteria and viruses. The characteristics of the antigen is that it can be a complete antigen or a hapten, meaning it's half incomplete. It contains antigenic determinants, and it can be a self-antigen. So antigens can be complete or incomplete. So a complete antigen means that it has two important functional properties. It has immunogenicity, meaning it has the ability to stimulate the proliferation of specific lymphocytes. So it can cause the mass production of a specific cell. And it has reactivity. It has the ability to react with activated lymphocytes and antibodies released by the immunogenic reactions. So examples of complete antigens are foreign proteins, anything that comes into your body that your body doesn't recognize, or polysaccharides, lipids, and nucleic acids. So anything that doesn't belong to you, which is what we see on a lot of foreign invaders like pollen and microorganisms. Incomplete antigens are also called haptens. These involve molecules that are too small to be seen, so they are not immunogenic by themselves. So examples of these are small peptides, nucleotides, and hormones. May become immunogenic if the hapten attaches to the body's own proteins. So the combination of the protein and the hapten is then seen as foreign. So this is going to cause the immune system to mount an attack that is harmful to the person because it attacks the self protein as well as the hapten. So this is what we see with poison ivy and animal dander and detergents and cosmetics. So antigenic determinants are the part of the antigen that the antibody or the lymphocyte binds to. Most naturally occurring antigens have numerous antigenic determinants that mobilize several different lymphocyte populations. They form different kinds of antibodies against them. These are large chemically simple molecules, or large chemically simple molecules like plastics have little or known immunogenicity. In other words, they can't trigger an immune response. So here is our antigen, the big red guy, and you see has lots of places that the different antibodies can bind to. Self-antigens, so all of your cells are covered with a variety of proteins. Those are located on the surface. They are not antigenic to self, but may be antigenic to others in transfusion or grafts. So you basically have a flag that says, I belong. And all of your cells in your body know that that cell belongs to your body. However, when you transplant tissue, including blood, if you don't have the right antigens, then you can create a reaction. All right, so that's what transfusion reactions are all about. That's what rejection reactions are all about. That's when you have the introduction of non-self cells and the immune system of the receiver sees those as foreign. One important group of cell proteins are the group of glycoproteins called the MHC proteins. What that stands for is major histocompatibility complex, and those are unique to you. So this is genetically determined. It is basically a flag that says this cell belongs to this person. It has a groove that holds a piece of the self-antigen or the foreign antigen. The T lymphocytes can recognize only antigens that are presented on MHC protein. So when we talk about the lymphocytes and their role and the antigen presenting cells and their role in adaptive immunity, there's three types of cells that we need to talk about. So there's actually two types of lymphocytes, the B cells and the T cells. The B cells are all about humoral immunity. The T cells are all about cellular immunity. And you will remember that they got their name based on where they matured. So they all originate in the bone marrow, but the B cells stay there and mature in the bone marrow. The T cells originate in the bone marrow, but they mature in the thymus, hence the name T cells. So antigen presenting cells are the other type of cells that are important in the adaptive immune response. These don't respond to specific antigens, but they play a role instead in immunity, an auxiliary role. Auxiliary role, that's the word I'm looking for. What about lymphocytes? So again, lymphocytes have a common development, and their steps in their life cycle is common. There's 5 general steps. Again, the origin for these is the red bone marrow. All right, so the origin for both the B and T cells is the red bone marrow. Then the maturation process. Lymphocytes are educated and mature in primary lymphoid organ, either the bone marrow or the thymus. This is where they gain immunocompetence, which is the ability to recognize only one specific antigen. B and T cells display only one unique type of antigen receptor on the surface when they are mature, and they can bind only one specific antigen. So you can think about the maturation process kind of like they go to school to be trained to do something, and they're very specific about what it is that they're trained for. Self-tolerance is the ability for the lymphocytes to be unresponsive to their own antigens. In other words, self-tolerance is your lymphocytes not attacking your own cells. T cells that mature in the thymus undergo both negative and positive selection pressures or tests. A positive selection process, this selects for T cells that are capable of recognizing self proteins. Those that are not able to are destroyed. In other words, these cells could potentially attack your own cells, so they're destroyed. So the ones that aren't able to recognize which cells belong and which cells don't belong are 86'd. Negative selection is what prompts apoptosis of T cells that bind to self-antigen that's displayed by the self cells. This process, called clonal deletion, makes sure that we have self-tolerance. So basically, either way, if you are not able to recognize a cell as belonging to the body, or if you accidentally or on purpose bind to the cell in the body, either way, you're going to be taken out of the process, because you could ultimately attack self tissue, which is the basis for autoimmune disease. All right, so it's showing you positive selection versus negative selection. So B cells are going to mature in the red bone marrow. Not much is known about this process. Only immunocompetent B cells are allowed to mature. So there's some type of selection process that happens for them as well that basically determines which ones are going to be self-reactive, and those are eliminated by apoptosis. So there's the idea. We had origin in the bone marrow. Maturation is going to depend on where they go to do their training and learn to be a good lymphocyte. All right, so again, back to this picture where we saw the origin in the bone marrow, then they go for maturation and training in either the thymus or the red bone marrow, and then once they have learned to do their job, then they go out and they populate the secondary lymphoid organs and in the circulatory system. Then finally, even though they know what they're supposed to do, they haven't actually come in contact with any antigen yet. So they have their first encounter and they become activated. So naive lymphocytes encounter the antigen that triggers a lymphocyte to develop further. Then the lymphocyte is selected to differentiate into an active cell by binding to its specific antigen, and this is referred to as clonal selection. If the correct signals are present, the lymphocyte will complete its differentiation into an active cell. So that's that final step that they're showing you, number 4 there. Then we have what's called proliferation and differentiation. So once the cell has been selected and it's been activated, the lymphocyte proliferates or makes copies of itself and forms an army that is exact copies of itself. They refer to those as clones. Most of the cells are going to become effector cells, and they're going to go out and they're going to fight infections. A few of them are going to stay back and become memory cells, so they're going to be able to respond to the same antigen when you come in contact with that years on down the road, the second time, third time, 100th time. These are the ones that are going to activate the immune system so that you don't get sick. B cells and T cells, B and T memory cells and effector cells are going to circulate continually. So there's that last step of proliferation and differentiation. So antigen receptor diversity, it's genes, not antigens, that determine which foreign substances the immune system can recognize. So you are genetically programmed with your immune system. So there are a variety of immune cell receptors that are the result of acquired genetic knowledge of microbes. So there are approximately 25,000 different gene codes for up to 1 billion different types of lymphocyte receptor antigen receptors. So that means we have a huge variety of receptors. The gene segments are shuffled around, and that results in lots and lots of combinations. So there are diseases that are defects in the immune system that are considered genetic. In other words, you didn't inherit the right genes to be able to fight certain pathogens. All right, so there's the overview of the B cells and the T cells in a chart form for you. What about antigen presenting cells, or APCs? These engulf antigen and present fragments of the antigen to the T cells for recognition. So there's 3 major types that we talk about. There are the dendritic cells, the macrophages, and B cells. So dendritic cells are found in your connective tissue and your epidermis. They act as sentinels, mobile sentinels. So they're just kind of cruising around in the boundary tissues looking for something that shouldn't be there. They phagocytose pathogens that enter into the tissues, then they enter into the lymphatics and they present the antigens to the T cells in the lymph node, and the T cell goes through its process. All right, so basically these go pick up things that shouldn't be there and present them. That's why they're called antigen presenting cells to a cell that can do something about them. Dendritic cells are the most effective antigen presenter known. This is the key link between our innate and our adaptive immune system, because these are considered part of our innate immune system as well. They're the ones that are patrolling around the connective tissues in the epidermis and picking up anything that they find. So there is a electron micrograph of one. What about macrophages? These are widely distributed in the connective tissues and the lymphoid organs. They present antigen to T cells, which not only activates the T cell, but it also further activates the macrophage. When a macrophage has become activated, it becomes extremely voracious or extremely hungry. So it becomes a very vicious killer. It also triggers powerful inflammation responses and recruits additional defenses once it's activated. What about your B cells? These are also considered antigen-presenting cells. So they do not activate naive T cells. They present antigen to a helper T cell to assist in their own activation. So let's talk about your humoral immune response, all right? So when the B cell encounters the target antigen, it provokes the humoral immune response. So it's going to cause the production of antibodies that are specific to that antigen that it has encountered. So B cells are activated when they bind antigen to their surface receptors and cross-link them. This triggers the receptor-mediated endocytosis, which we learned about last semester. Remember that specific endocytosis of cross-linked antigen receptor complexes through the process of clonal selection, which leads to the proliferation and differentiation of B cells into effector cells. Most clone cells become plasma cells, and plasma cells are the ones that are going to secrete the antibodies that are going to attack the antigen. So we secrete specific antibodies at a rate of about 2,000 molecules per second for 4 or 5 days, and then they stop doing this. Antibodies circulate in your blood and your lymph, and they bind to free antigen, and they mark them for destruction by innate or other adaptive mechanisms. So how does this one work? So we're talking about the B cell side here. So T cells that we'll see, T cells are actually going to actively hand-on-hand combat with antigen. But B cells, they don't like to get their hands dirty. I like to think about it like that. So our antigen are our little red things, and they bind to a B lymphocyte, right? And the B lymphocyte is going to proliferate to form a clone, which means that they become activated, and the clones become the plasma cells, and the plasma cells secrete antibody, and it's the antibody that's going to actually bind and attack the antigen. All right, so B cells do this whole thing without getting their hands dirty. Clone cells that don't become plasma cells become memory cells, just like we saw a few minutes ago, and those are going to provide immunological memory and mount an immediate response should you come in contact with that particular bad guy again. So there's the idea of the plasma cells. The plasma cells are secreting antibodies, but there are a few that become memory cells. So let's say you had chickenpox, right? You secreted antibodies for that, and then you made memory cells. Those memory cells were stored away, and surely you have come in contact with chickenpox numerous times since that occasion, but because the memory cells are already primed as soon as that antigen was detected in the body, it causes the same response of the same antibody release and attacks the antigen very quickly, and you don't get sick. Primary immune response is when you have the proliferation and differentiation after you've been exposed to an antigen for the first time. This has a lag period of 3 to 6 days. You reach the peak level of your plasma antibody in about 10 days, and then the antibody levels start to decline. Secondary immune response is when you come in contact with that same antigen later. It could be months later, it could be years later, but you have an immune response, but it's much, much quicker, and therefore you rarely get sick in that event. All right, so your sensitized memory cells are providing what's called immunological memory. This can respond within hours, not days, so that you don't have to have that lag period. The antibody level is going to peak in 2 to 3 days at much higher levels, and the antibodies bind with much greater affinity, so it's a stronger bonding between them. The antibodies levels can remain high for weeks to months. So there's the idea of your primary response, and then what you see happen with the secondary response. Active humoral immunity occurs when B cells encounter the antigen and produce specific antibodies against them. There are 2 different types of active humoral immunity. Naturally acquired means that you actually came in contact with the bacteria or the virus in some way. Artificial means that you were exposed to it through a vaccine of either dead or attenuated pathogen, and attenuated means severely weakened to the point where it can't make you sick. These are going to provide the antigenic determinants that are immunogenic and reactive, and that makes it so that we don't have to go through the primary response or experience the illness. Passive humoral immunity occurs when the antibodies are introduced into the body. So when we're talking about active, we're making the antibodies ourselves. We're either doing it because we were exposed to the disease or because we were intentionally exposed, like through a vaccine. Passive, we're not going to make the antibodies ourselves. In passive, what we're going to do is we're going to get ready-made antibodies. All right, so the B cells are not challenged by the antigens, immunological memory does not occur, and protection ends when the antibodies degrade. So there are two types of passive humoral immunity. There's naturally acquired, and this is what you see happen when mom is passing her immune system over to the fetus during pregnancy through the placenta or because of breastfeeding. So when mom is breastfeeding, she is passing over her immune system to her baby. So that's why we want to encourage mom to breastfeed for as long as possible. And then we have artificially acquired. This is where we're getting an injection like a gamma globulin shot that is giving us the antibodies that we need. So in some conditions like hepatitis A, it would take your body about 6 weeks to ramp up to the peak levels of antibodies that you need to fight the virus. And in that 6 weeks, it could do a lot of damage. And so we give you, through a gamma globulin shot, the antibodies to bridge the gap until you make them yourself. So that's what artificially acquired passive immunity is. It means that we've given them to you, but realize that the protection ends when the antibodies are naturally degraded in the body. But hopefully your own immune system is picked up by that. So there's the two sides. All right. Active, naturally acquired means that you came in contact with it and you got sick. All right. Artificially acquired means that you got a vaccine of either dead or attenuated pathogens, so you don't experience the illness, but you're protected from it. All right. In passive, naturally acquired, we're passing the antibodies. So there is the key word. In passive, we are giving the person the antibodies that they need to fight the illness. They're not making them themselves. So the antibodies are passed from mother to fetus, either through the placenta during development or from mom to the baby through her breast milk. And artificially acquired is when we are actually giving you exogenous antibodies in the form of gamma globulin to make sure that your body is able to fight the illness adequately. So antibodies, I've been talking about them for a while now. They're also called immunoglobulins, and that is where their abbreviation comes from. So IgSs are proteins that are secreted by the plasma cells. They are made up of a gamma globulin portion of your blood. So that's where they are derived from. They are capable of binding specifically with an antigen, and they are detected by that's detected by B cells, and they're grouped into one of 5 classes. So the structure of these, overall, they have a T shape or a Y shaped antibody monomer that has 4 looping polypeptide chains that are linked by disulfide bonds. So basically a lot of verbiage that says that we have 4 pieces of amino acids, chunks of amino acid that are hooked together by double sulfur bonds. 2 chains they have 4 chains. 2 of them are identical heavy chains with a hinged region in the middle, and 2 are identical light chains. And then there's a variable portion at the end of each arm that combine to form 2 identical antigen bonding sites. The stem makes up the constant region. So this is the area that determines which antibody we're talking about, and this is considered the effector region of the antibody. It serves a couple of common functions in all antibodies. It dictates the type of cells or chemicals that the antibody can bind to and how the antibody class functions to eliminate the antigen. So we're going to talk about what are the different ways that we get rid of this antigen. So there's a picture of what one looks like. So you got a heavy chain and a light chain. So the blue and the purple on each of those, and then you have the stem region. And there is a model of it. So let's talk about the classes, because that's way more important to me than the structure of it. So you have 5 main classes: IgM, IgA, IgD, IgG, and IgE. And so notice that it's capital I, little g, and then a big capital letter. So that is how you properly denote that. So IgM is a pentamer, and it's larger, so it's larger than the rest of them. It's the first antibody that's released. That's something notable to know. It has potent agglutinating activities that it can conduct, so it's going to make things clump together, and it readily fixes and activates complement. So that's how this particular one works. IgA, also called secretory IgA, is a monomer or a dimer. It's found in mucus and other body secretions like tears, saliva, those kinds of things, and it helps to prevent the entry of pathogens. So we find IgA in all of the openings of our body. And so there's the picture of them so that you can get an idea. Then over on the right hand side, all the verbiage I was just talking about. You want to make sure you know all of the important little things about them, like the fact that IgM is the first immunological class that's secreted by the plasma cells, and that it fixes and activates complement. IgD is a monomer that attaches to the surface of B cells, and it functions as a B cell receptor. IgG is a monomer. It is most of the antibody that you find in plasma, 75 to 85%. So it's the most abundant one. It's from secondary and late primary responses. This is the one that has the ability to cross over to the placenta. So this is the one that mom's passing over to her offspring. And then IgE is the monomer that is active in allergies and parasitic infections. And what it does is it causes the mast cells and the basal cells to release lots of histamine. Plasma cells can switch from making one class of antibody to another. So we didn't see this very many places, but this is one place where a plasma cell has the genetics to make different ones. It retains specificity for the same antigen in the process. So IgM is released during the primary response, but the plasma cell can switch over to IgG for the secondary response. Almost all secondary responses involve IgG, and switching from IgM to A to E can also occur. So there is D, G, and E. So how do they do their job? So antibodies are going to target antigen, but they don't destroy the antigen. They inactivate it and they tag it for destruction. And when they bind to it, they form what's called the antigen antibody complex. All right. So at the end of the day, the result is the same. We're going to get rid of the antigen. It's just how do we do that? We can do that through neutralization, agglutination, precipitation, or fixation of complement. So neutralization is the simplest, but one of the most important defensive mechanisms that we have. The antibodies are going to block several sites on the virus or the bacterial exotoxin, and they prevent the antigen from binding to the receptors on the cell tissue, and the complex undergoes phagocytosis. So we basically neutralize it. We get rid of it. Agglutination, the antibodies can bind the same determinant on two different antigens at the same time. Each antibody has two arms. Each contains a variable region capable of binding one antigen. This allows for antigen-antibody complexes to become cross-linked and forms a big chunk or clump. All right, so we call that process agglutination. This is what you see with mismatched blood cells. So basically, we've clumped the bad guys together so that we can get rid of them. Precipitation is when we have soluble molecules instead of the cells that are cross-linked into complexes. The complexes precipitate out of solution. They basically rain out of solution, and then the precipitated complexes are much easier for the phagocytic cells to engulf and destroy. And then we also can have complement fixation and activation. The main antibody defense against your cellular antigens like bacteria and mismatched red blood cells is complement fixation and activation. When several antibodies are bound close together on the same antigen, the complement binding site on their stem regions are aligned, and this triggers the fixation of complement. Remember the 20 proteins that are floating around in your body in the inactive form, which can lead to cell lysis as well as other functions. For example, it can cause an amplification of the inflammatory response or sugar coating opsonization. So there is the big picture idea. We form the antigen-antibody complex with the antibody, and in some way we get rid of it. So it shows you those 4 different ways and ultimately the fate of the antigen. So parasitic infections by worms like Ascaris or Schistosomonas require a different attack plan. So when we have parasitic worms that are too big for the regular precipitation, lysis, agglutination, neutralization, what they call plan attack, what we just talked about, we have a different way we go about doing this. So IgE antibodies still are going to play a critical role in the destruction of the worm. They bind to the surface of the worm. They mark it for destruction by the eosinophils. So if you remember back to eosinophils, if you see an elevated number of those, it indicates that your patient has a parasitic worm infection or could indicate that. So now you see why, right? So the IgE is going to attach to the surface of the worm, and then that causes the proliferation of eosinophils, which is why you see the elevated number. The eosinophils bind the exposed stems of IgE, which triggers the eosinophils to release their toxic contents, which basically dissolves or lyses the worm from the outside in. We use monoclonal antibodies as clinical and research tools. So monoclonal antibodies are commercially prepared pure antibody that are specific for a single antigenic determinant. These are produced by hydroomas, which are cell hybrids that are formed by the fusion of a tumor cell and a B cell. So the tumor cell allows the cell to proliferate indefinitely, and the B cell part of it is going to allow for the production of the antibody. So basically, we genetically modify the tumor cell so it's not dangerous anymore, but it just still has that ability to massively proliferate, which is the hallmark of a tumor cell. We use these in research, clinical testing, and for cancer treatment. So what's the summary of antibody actions? The antigen-antibody complexes don't destroy the antigens. They simply prepare them for destruction by the innate defenses. The antibodies go after extracellular pathogens. They don't invade into solid tissue unless a lesion is present. There is a recent exception to this. Antibodies can act intracellularly if they are attached to a virus before it enters into the cell, and then they can activate mechanisms that destroy the virus. So there's the end of part 2. I'll see you in a few minutes for part 3.